Welcome to HudsonAlpha. My name's Sarah Sharman. I'm HudsonAlpha's Research Communication Manager and one of the voices of HudsonAlpha's podcast, Tiny Expeditions. I'm so excited to welcome you to this remarkable event where two of our faculty investigators will explore the intersection of science, technology, and humanity and offer hope in our fight against global food insecurity. First, we would like to thank our sponsors, Sandy and Bob Hovde, for sponsoring tonight's event. This program is part of the larger 15th anniversary seminar series, which is sponsored by Avolution and the Keen Group of Morgan Stanley. We would like to thank you all so much for your generous support of HudsonAlpha. Now, if you could imagine with me a world where hunger is a distant memory. A world where everyone, regardless of their age, race, geographic location, or circumstances, has access to sustainable and nutritious food. Sadly, this is a distant dream for far too many people in our world today. Around the globe, food insecurity haunts hundreds of millions of people, casting a shadow on their lives, limiting their potential, and depriving them of one of their most basic necessities for life. Now, if you're a curious person like me, you're probably wondering why, in a world where technological advancements seem limitless, we are still struggling with a problem like this so fundamental. Unfortunately, many factors complicate the answer to this question. Our climate, which was once predictable and dependable, is now erratic and volatile. Temperatures are increasing like we've never experienced before. Weather patterns are shifting with more extreme weather events every year. Crop plants and the very plants that help sustain our environment are struggling to adapt. Our population continues to skyrocket, stretching the limits of our resources and placing an unprecedented strain on our ability to feed the masses. With more people comes a larger carbon footprint. This puts an additional stress on existing plant ecosystems. In this vicious cycle of interconnected issues, it's apparent that we need a solution that harnesses the very essence of life itself, genomics. Genomics holds immense promise in transforming how we combat global food insecurity. It offers a key to unlock the secrets of nature, unraveling the genetic code of plants and animals alike. With this knowledge, we can tailor plants to thrive in inhospitable conditions, resist disease, and yield bountiful harvests. We can create a sustainable, resilient food system that will nourish us all, even in the face of adversity. Tonight, Drs. Clevenger and Harkess will share stories from their pursuit of a world where food is no longer a privilege, but a universal birthright. We will hear about cutting-edge research, technological breakthroughs, and the unwavering dedication of scientists, farmers, and visionaries who refuse to accept a world where hunger reigns. We hope tonight's discussion will challenge your perceptions, captivate your imagination, and instill a sense of urgency in your hearts. So please, sit back and relax, and turn your attention to the stars of tonight's show, HudsonAlpha faculty investigators Josh Clevenger and Alex Harkess. Together, they will reveal the potential of genomics to reshape the future of agriculture and eradicate food insecurity from our planet.
Within each of us, there is a book. Every single one of you has a story to tell about where you came from and where you are going. For that exact same reason, I fell in love with plants. They are marvelous storytellers about the history of our planet. Plants tell us stories about diversity, about adaptation, about change. They tell us stories about survival on a planet that is not always so hospitable to life. When I think about the history of plants on our planet, I'm charmed. The first green organisms started photosynthesizing nearly 1 billion years ago, pumping oxygen into our atmosphere that made it livable for us today. The first plants moved on to land about 500 million years ago. and shortly after began setting their roots deep in the soil, strengthening the earth. Nearly 130 million years ago, the first flowers began to evolve and bloom and blossom and fill our beautiful planet with color. But we, as humans, as a species, have only been farming plants for roughly 12,000 years. It's the blink of an eye relative to other organisms that have been living on our planet. It might be strange to think, but an oak tree has seen far more of our planet's history than you or I ever will. And that scares some people, to realize that we as humans are a short-lived species, that trees will live longer than you will. But instead of being afraid of it, I like to view it as an opportunity, as a responsibility to take the short time that we as humans get to live on this planet, to improve it for this generation and for all of those moving forward. So what can we as humans do to learn from plants that have survived on our planet for the last billion years since the first green thing evolved? Well, luckily, we share a common language, which is helpful because plants cannot tell their story for themselves. We all share the language of DNA. 4 letters, A, T, C, and G, that unite nearly all things that are alive on our planet today. HudsonAlpha is a unique place in this sense. This building that you are in right now, this whole campus, is not just a research institute. It's much like a printing press. We take DNA from any organism on the planet, and we can put it inside of a printing press, what we call a genome sequencer, and we can write words from those letters, words that we call genes. We can take those genes and put them into the right order, like words in a chapter. We just call them chromosomes. We can take those chapters and put them all the way into a full book that we call a genome. And once you have a book that represents all DNA sequence for an organism, you put it into a library. And we start to organize this beautiful diversity that we've created from these genome sequencers. And when you have a library that's filled with hundreds of thousands of books that represent diversity of DNA across the tree of life, and you have a librarian who allows you to ask questions about those books, we begin to ask some of the most fundamental questions that can change the history of our planet. Are there genes inside of that library that allow us to grow more food on our planet? Are there genes in that library that allow us to grow plants that use less water, that need less fertilizer? Are there genes in that library that allow us to farm plants in new places that we previously had not been able to? Can we use genomics to find tiny genes that have big effects?
I'm so happy right now. I'm sitting here looking at all of you and listening to this guy, and I see my kids arguing. And what a joy. What a joy for me. I really made it that my kids can sit here watching me talk to you. So we're going to take you through 4 incredible stories about how tiny genes have had big effects across the world. Things that we both have worked on and that we're working on here at HudsonAlpha, and hopefully give you hope of the future of food security. But the first thing that we have to do is we have to talk to you about why agriculture is on the brink. Now, it's not on the brink, but it's always on the edge. It's always on the edge of not producing enough safe and nutritious food for us and for our fellow humans across the world. And there are 4 major things that crops have to produce those safe and nutritious food. In the threat of all of the pests and all the viruses and diseases that are always threatening them, they have to have these 4 major things to be healthy. And those 4 things, if we start from the top and go to the bottom, can start with the climate. And the major component of climate is the sun. And we know that the sun has been harnessed by plants to turn that light into energy in the form of glucose. But it's not just that. Plants use the sun, just like us, to trigger their reproductive cycles, their processes. How much light they get at a certain time tells them, oh, it's time to germinate, it's time to grow, it's time to make flowers and not vegetative tissue. The amount of sun tells them all of these things and gives them those triggers on when they need to do them. But also a part of climate is temperature. So different plants need different temperatures at different times. Long stretches of cold temperature tells that plant that, hey, when this is over, it's time to germinate and to grow. It needs to be warm enough for that tiny plant to thrive in that environment. But instead, at the end, it has to be warm enough for the grain and the fruits to mature so that we can harvest them and use them for food. And so as climate changes, different plants interact with those changes in different ways, in different magnitudes. And there's always that chance that the crops that we rely on cannot provide the food that we need in the face of those changes. And so if we're moving down, the second thing that the plants need to thrive and be safe and to be healthy is water, of course. And water is essential to life. And plants, it's the same way. So it provides that electron that is donated in the process of photosynthesis to produce glucose for life, for all of us. And it also carries nutrients from the roots to the leaves and from the leaves to the roots and makes sure that the right things are in the right tissues at the right times. And as well as providing turgor for the cell so that plant could be rigid and grow in the environments that it would like to. But it's not just water, it's fresh water. It's water that has the right amount of minerals to interact with the plant cells for it to be healthy. And so recent studies suggest that by 2030, there'll be 40% more demand for fresh water than there'll be supply. So now more than ever, we have to grow more crops to yield more in the face of less water and less resources. And so the third thing, one of the nutrients that the water transports throughout the plant is nitrogen. And nitrogen is the main player in chlorophyll, the engine that uses the sunlight to produce energy. It's also the building block of amino acids, which are the building blocks of proteins. Now, some plants fix nitrogen. Those are ones that I like, particularly peanuts do that. But most of our crops do not, and we have to add nitrogen on them for them to grow and be healthy. Now, unfortunately for us, they're not as efficient at uptake of that nitrogen and utilization of that nitrogen, and so much of that nitrogen we add to fields provides substantial pollution in our waterways and in our air. And not only that, but synthetic fertilizers, the Haber-Bosch process, which revolutionized the 20th century and kickstarted the Green Revolution, needs a lot of energy and a lot of other resources, and global perturbations have a major effect on the availability of that fertilizer. And just last year, 2 years ago, fertilizer prices doubled overnight. And those of you that know growers or grow things yourself know that that was a major, major challenge for those folks that are trying to produce our food. So the fourth major thing, and obviously there are more things, but these are things that we must have, is healthy soils. And we now know that healthy soil is not just a place to lay down roots, it's not just a safe harbor for that water and those nutrients that the plant needs, but it also is a thriving ecosystem that when it's at most healthy, provides amazing resources for that plant. So one of the easiest things to think of in the microbiome is those bacteria and microbes that are interacting with the plant in this elegant dance and providing nitrogen, where the plant is sending out signals and saying, hey, come live with me. I will provide you vitamins and minerals and sugars, and you will provide me with nitrogen. But also, amazingly, recently, we've discovered that in many cases, this healthy microbiome, this collection of microbes that are living in the soil, actually provide this community defense against many pests and allow our crops to thrive. And so, in the face of all of those things that the plants need, that we need to grow those plants, there are many challenges. And global crops, crops that are major commodity crops, crops that are regionally very important, they all are affected in different ways by these different aspects of need that they have to have. And so now more than ever, to feed ourselves, to feed our fellows, we have to be better, faster, and more efficient at understanding how genetics can be used to help those plants thrive. So I want to end on this first part, this introduction, to tell you that in 2020, an estimated 800 people around the world went hungry. That's 1 out of every 10 people on the planet. An estimate 45 to 50 million more people every year will be added. But I want to ask you a question to think about as we talk tonight. In the face of all those challenges and all of those things that we need to have safe and nutritious food, would you believe me if I told you that a single gene helps save 1 billion lives?
I was never supposed to be a plant biologist. Sometimes I wonder how I got here at all. I thought I was going to be a lawyer, actually. It's hilarious thinking about it, actually. And on my first day of college at Miami University of Ohio, I meet a botany professor who spent 30 minutes with me and changed the course of my life. A guy named Jim Hickey. He studied ferns, discovered new species of ferns out in the Bahamas in particular, and he said one sentence that has stuck with me ever since my first day of college. He said, "Alex, botany is the study of history and nothing else." He helped me get into a laboratory studying papaya plants. And I still remember the first phone call home to my mother. My whole family's medical, they're all orthopedic surgeons, and I said, "Mom, I'm studying papayas." And she said, "Why? What's interesting?" And I told my mother, "Well, I've learned a lot about the power of one gene in papaya plants." In the 1940s, papayas were grown all throughout Hawaii, and still are, of course. It was one of the most nutritious, high vitamin A, high vitamin C. You grow them not just in Hawaii, but across India and beyond. An incredibly nutritious crop grown worldwide at this point. But in the 1940s, an immense challenge started to rear its head. Papaya ring spot virus is a virus that began taking hold across Hawaii, and within the next few years, nearly 50% of the crop in Hawaii of papayas was infected by this virus, that once you get it in your field or your farm, there is no escaping it. You can only run from it. And on a place like Hawaii, there is only so much land to run from. So what did they do? They had to come up with a solution, the growers and the scientists, because this was a major industry across Hawaii. Right? You can't lose papayas because all of the farms and the farmers relied on it. People relied on it for nutrition and for calories as well. Biotechnology was the solution. They took one gene, the coat protein from the ring spot virus, and they inserted it into a papaya cultivar. And it effectively primed this papaya cultivar to already recognize this viral pathogen. So it mounted a defense before it even saw a virus for the first time in its life. One gene that was inserted into a papaya plant solved all susceptibility problems, gave full resistance to papaya plants across the island and beyond. One gene with huge effect. But that's not the only problem that papayas face. Nowadays, my laboratory studies plant reproduction, and again, as it often goes, one conversation with one professor changed the course of my life. This papaya professor that I worked with explained to me that there's a reproduction problem in papaya plants. And just like some mammals and animals, papayas have multiple sexes, actually. So when you grow up a papaya plant, it could either be a male plant that makes male flowers, it could be a female plant that makes female flowers, or it could be a hermaphrodite plant that makes flowers that have both male and female parts inside of them. The challenge is that male plants don't make fruits. Female plants do make fruits, but they're nice and circular, but they don't taste good. And hermaphrodite papaya plants are the only ones that make those nice, oblong, football-shaped, delicious papayas. But here's the challenge. When you grow papayas up from seed, you don't know if they are male or female or hermaphrodite until at least 9 months until those plants grow big and tall and make their first flower. So imagine you're a grower. You're growing lots of papayas from seed, and you're fertilizing them. You're using water, you're using energy, labor, space, time, especially on an island like Hawaii that is limited in terms of space. It is at a premium. Well, is there a way that biotechnology could solve that problem? Do we need to grow all 3 sexes of those papaya plants, or can we identify the genes that control whether a papaya plant is a male, a female, or hermaphrodite? Or could we screen for it early on before you spend all of that time and energy and passion and love as a farmer growing up plants to be huge before you eventually rip them out of the ground? This was 15 years ago when I was an undergraduate. We didn't have the tools like we have here today at HudsonAlpha. Just like the Human Genome Project revolutionized the way we think about human disease and development, this was before that for plants. So I bided my time. But I was obsessed with the idea that if we can control reproduction in plants, you can control food. Because at the end of the day, all of global food production relies on the very simple fact that plants make flowers, flowers make pollen, and pollen moves from one plant to another to make fruit. That has absolutely captured me as a scientist. That basic fundamental process that is so challenging to understand and comprehend, that is what my laboratory here at HudsonAlpha works on. I went on to do my PhD at the University of Georgia. I made another phone call home to my mother and said, "Mom, I'm studying asparagus." And I promise the next time you go to the grocery store, you are going to see asparagus plants and you are going to think of me for the rest of your lives. Because when I was a graduate student, asparagus had the same issue as papayas, except 50% of the seeds would be male and 50% of seeds would be female whenever you grow asparagus plants up. The challenge is that breeders and growers only want male asparagus plants because an asparagus plant, a single one, can live 30 years or longer in a field, and females make fruits that drop their seeds, and all these seedlings pop up in the fields, muddying up your nice, tidy rows of a perennial crop like asparagus. Males also seem to live a little bit longer and maybe taste a little bit better too. But it's even worse than papayas, because when you plant an asparagus seed, it might take up to 3 years before you know if it's a male or a female. Wasted time, water, fertilizer, energy, space, love, passion, all of it. Is there a solution that we could come up with? Well, I'd waited a few years since my undergrad days working on papaya, and genome sequencing was starting to become more powerful, more usable, more applicable to plants. We took what they did with the Human Genome Project and we brought it into the plant biology world. We generated a genome for asparagus, just like the Human Genome Project. And being a plant biologist is exciting because you can do things that would be wildly unethical in humans. We took tens of thousands of asparagus seeds that should have all been males, and we threw gamma rays at them to create large deletions in chromosomes, small deletions, big inversions, translocations. We messed the genome up. And the idea was, well, can we potentially knock out or mess up some of these genes that make an asparagus plant a male or a female? Can we find the genes that control whether a plant is one sex or the other? And the answer was yes. We got lucky, actually. We found 2 genes side by side, very close to each other. But when I modify one or both of those genes together, I could control whether an asparagus plant was a male, a female, or nothing in between, or a hermaphrodite. This basic idea allowed us to change the asparagus industry because no longer did you have to wait 3 years to understand if a plant was going to be male or female. Now you knew right away from a seed. We effectively were able to double the yield of asparagus plants in a single plot of land because you did not grow 50% male and female, you grew 100% male. Time, water, energy, and more. What we were able to do is to find tiny genes that had big effects.
So, when I was an undergrad, I tried to be in a punk rock band. I called my mom and I said, "Mom, I'm studying peanuts." And she said, "Oh, thank God." I thought of that just now. That's a great joke. So when plants moved across the world, they took their pests with them. And like the diseases that we have, some can be endemic to a certain area, but the most devastating tend to be shared across the world. And so in science, some of the most profound effects happen unintentionally. And that's the story I want to tell you right now. So in the 1960s, a young PhD student at North Carolina State University spent 17, maybe 25 hours a day in the lab looking at chromosomes under a microscope. He told me that he never slept and he never ate, and he did that for 4 years straight. I don't think I believe him, but he insists that he did. He was just interested in understanding how chromosomes interact when you take two species that have different numbers and you pair them. So how might they interact in the cell? That's all he was interested in. And he was looking at peanut lines that took 15 years to develop by his advisor. Now think about that. For 15 years, they went through this complex process and looked at plants every year under the microscope to see, are these viable peanuts? Because they had crossed a wild peanut species that had 20 chromosomes with a cultivated peanut species that had 40. So after all of those years, they had viable seeds, and he did his PhD, a wonderful PhD on lots of great pictures of chromosomes and how they might interact. And the peanuts were growing outside. And in North Carolina, where all the peanuts are grown that you buy and eat at the ballpark, the Virginia ballpark peanuts, they didn't have problems with some of the most devastating fungal diseases of peanut, late leaf spot and rust. And so these lines were fine. They weren't particularly useful for anything. They grew fine. Nobody liked them very much. They had really dark red skins. But there was a visiting researcher from India, and he looked at them and he said, you know, I'm really interested in these because they may have some wild species segments in them. And so he took them back to India. And unbeknownst to everybody, one of those lines that was named CS16 had inherited two small segments from the wild species, a very small set of tiny genes that doubled the resistance genes in that plant. And so when he took them back to India, what did he find? That they were practically immune to rust, a disease that was devastating the entire country. And so he took those lines and he bred peanuts with them that would thrive in the country, and he released a peanut variety that was immune to rust and had great late leaf spot resistance, and it revolutionized peanut production in the country. And something odd happened. They changed the names in the pedigree. So nobody knew that those lines came from North Carolina. They had no idea where they had come from. And so colleagues from all around the world said, I want those seeds. And they took them back to their countries, and they bred varieties that were resistant to rust and late leaf spot. And all told, 30 countries produce resistant varieties, representing over 90% of global peanut production. This went unknown for close to 30 years. All around the world, growers were growing these lines and having that security that when devastation hit with the pests, that they would not have to worry about it. And so when we sequenced the peanut genome, which happened right here at HudsonAlpha, that was the first time we could find the answer to this question. And so when we looked at these resistant lines, because they were very, very resistant, we identified small regions that looked different, that had signatures that were different enough that they caught our eye. And so through comparative genomics, we identified those small segments actually came from a wild species that had been known to be resistant to many diseases. And armed with that new knowledge, we went across the world and collected DNA from all of these varieties that were resistant, and we looked for these sets of tiny genes. And you know what? Every single one of them had them in it. And so now having those tools for the first time, last year, peanut varieties were released in the United States that include this set of tiny genes. And in a 2-year economic study, it was shown that farmers could have 50% more net revenue spraying 0 fungicides when compared to a gold standard variety with a full fungicide spray regimen. This tiny set of genes had big, big effects.
If you were to go to the grocery store tonight, you might not see asparagus because it's seasonal. I know you'll think of me next time you do, though. But you will typically always find an apple. Have you ever wondered why you can always find an apple in the grocery store? Audience participation. Can you raise your hand if you have eaten an apple in the last month? Wow. And it was probably a deliciously sweet and incredibly fresh apple that was picked right off of a tree, correct? That apple that you ate had probably been sitting in a cold, refrigerated box for the last 9 months since it was picked off of a tree. The apple growing season is only 3 months old, and apples have this amazing ability to almost go into homeostasis. When you pick them off a tree at the right time, they can go into a refrigerated box, you drop the temperature, and they can be stored for up to a year without any loss of flavor or taste or texture or nutrients. It's an unbelievable aspect to a fruit like that. But here's the challenge, is that if you don't pick that fruit at the exact right time, it doesn't stay in a box forever. And in fact, if you pick an apple a little bit too soon or a little bit too late, even 1 week off on either direction, much like an angel share in a whiskey barrel, that apple rots away in a box over the next year. And all of that time and love and effort and water that went into harvesting that apple goes away. But let's think about what an apple actually is. As an apple is growing and developing after it's fertilized inside of a flower, well, it's still connected to a tree. And just like a baby connected to mom by an umbilical cord, when you cut that cord, or you pick an apple off of a tree, everything stops. All development, all ability for that apple to get a little bit bigger, to keep on changing, is over. And the challenge with being an apple harvester is, how do you pick an apple at exactly the right time? Not too early, not too late, so that it can sit in a box for up to a year before it gets to someone like you. Well, one way that we try and attack this question is through collaboration with folks like the United States Department of Agriculture, Washington State University, one of the greatest apple-growing regions in the country up in the Pacific Northwest. Well, what we're working on right now is asking if we can develop a biomarker, some sort of method using genetics to identify the perfect time to pick an apple off a tree. But an apple is an incredibly complex thing, actually. And it's almost like an orchestra playing a beautiful symphony. An orchestra that has thousands of musicians playing different instruments at different times, at different volumes, and crescendos going up and down. Just like that orchestra playing, plants have genes, and genes are turned on and off at different times of the day, different times of development, different kinds of stresses, whether that's water or beyond. And the big question that we try and ask as geneticists is, can we look at just one of those orchestra players, just one violin player, just one gene, and turn off everyone else? Turn off all the noise and listen to one person playing a note and ask, how loud is that violin playing? When does it turn on and off? How highly is that gene expressed? When does it turn on during development or the day or stress? This is the idea behind developing a biomarker, a signature, some way that we can use a genome sequencer to sequence these genes turning on and off in this beautiful orchestra that we call plant life, and identify gene or genes that perfectly allow us to pick an apple off a tree at the right time so that it sits in a box for a year. So what we've been doing with Washington State University and the USDA and the Washington Tree Fruit Research Commission is thinking about development in apples. So we take apples from all different maturities, from young to way too ripe, and we sequence their genes. Just like those orchestra players, we sequence all genes that are turning on and off at all times of maturity and development. And we try and ask exactly when we should pick that apple off a tree. But we're not the first people to do this. For the last 60 years, there's been a really classic test called an iodine test that's used in the apple harvesting world. As apples ripen, they convert starch to sugar. So you can use a starch test to ask, is this apple filled with starch, or is it beginning to ripen, lose its starch, and convert to sugar? And once it's starting to convert to sugar, or almost fully converted, that's when you pull it off a tree. The challenge is that there are hundreds, if not thousands, of different apple cultivars that were all bred to be different in different ways. So not every apple develops the same at the same times with the same levels of starch and sugars. And this is part of the beauty of eating apples, is that they are all so different, the ways that they look and taste and grow. But when I look at an apple, I think about it from a genetic perspective. And luckily, most apples that we look at share the same sets of genes. So even though they might mature and develop very, very differently, they have a core set of genes that allow them to reproduce and mature and grow. So when we look at these genes that are turning on and off along the time course of an apple developing, from different cultivars like Granny Smith and Honeycrisp and Cosmic Crisp and more, we're asking these questions to harvesters. When would you pick this apple off a tree? When would you decide that the starch test is perfect? And then we look at the genes, and the genes that are turned on and off at exactly that time. And what we've been able to do is identify a small set of genes that are highly predictive of when you should pick an apple off a tree. And I know that might be great for us sitting in this room, but the implications are huge. Right now, there are roughly 3.7 trillion apples every year that end up rotting in a landfill. Up to 50 to 100% of apple lots that go into cold storage, despite being picked at what they thought were the right time, could end up going entirely rotten. You might have heard of food deserts in our country. At this very moment, there are nearly 50 million people who don't have access to fresh produce in our country. Partly because produce doesn't exist in grocery stores that are nearby. Produce goes bad in those grocery stores. People don't have access to grocery stores to begin with. So what if something as simple sounding as an apple that can survive in cold storage for up to a year, that is highly nutritious, could be part of that kind of solution? Can we change the way that we think about food distribution, food access in our country? We are finding tiny genes that have big effects.
Finally. We were hoping you would clap so when we walked up it wasn't awkward. That was good. Congratulations to Alex and his wife Katie. What an amazing thing for him to give that speech and to have his son laud him the entire way. This is really great. There is a food contaminant that over 5 billion people in the world are chronically exposed to. This contaminant is so carcinogenic that it's attributed to 25% of new liver cancer cases every year. Not only that, but when you combine that with chronic hepatitis B infection, something that the developing world knows a lot about, you increase that by 30 times the risk for liver cancer. How many of you sitting out here, by a show of hands, have heard of aflatoxin? That's more than I was expecting. Some of you because you know me. But normally when I ask people that, that's what I get. Not many people have heard about it. And I've been thinking about how genetics can be used to alleviate the effects of chronic exposure to aflatoxin contamination for more than a decade. But before I heard about it, I had never heard the word before, nor knew of the profound effect it had on people's lives across the world. Now, aflatoxin is produced by fungus Aspergillus flavus. Aspergillus flavus loves the fats and sugar in nuts and grains that we also love. And it produces aflatoxin in the face of oxidative stress, so heat stress and lack of water. And we know that in those conditions, it is always going to produce it. Now, what sort of makes this hard is that aflatoxin has no negative effects on that plant's ability to reproduce. And so if you think about selection and evolution, there is no selective pressure for that plant to evolve any defense against the production of aflatoxin. Now let me throw you a curveball. What if I told you that even though this is one of the most important food security threats that we face across the world, there is not a single source of genetics anywhere grown that helps to mitigate aflatoxin? Now, we're a little bit of an impasse here, because the theme of tonight is we're going to find tiny genes that have big effects. So you might be thinking to yourself, well, where's he going with this? It seems pretty dire. But nature always gives us gifts, if only we know where to look. And so there's a walnut variety named Tolare, and scientists in California at the USDA noticed that in years where most of the walnuts had very high levels of aflatoxin contamination, Tolare never did. And so through a set of elegant experiments, they sought to find out why that was. And what they found was that in the skin of the walnut, what we call the pellicle, there was more than 2 times the amount of tannins. The tannins that are in your wine, the tannins that are in your green tea. And they saw that those tannins, that was the thing that was causing the suppression of aflatoxin. So it turns out, in this wildly odd twist of fate, that the fungus itself has enzymes that hydrolyze those tannins in the seed of the walnut, producing gallic acid. And those researchers found that it was the gallic acid and nothing else that completely suppressed aflatoxin. And so Tolare walnut are effectively immune to contamination. So why is that? Why would that be? Well, remember when I told you that aflatoxin was produced in the face of oxidative stress? Well, there are 2 very sort of easy hypotheses that we can come up with. One is that the gallic acid itself is alleviating that stress through meditation. No, I'm just kidding. Through the scavenging of reactive oxygen species to create an environment that's not stressful. Or, as aflatoxin is produced and controlled by a master regulator that is also turned on by oxidative stress, the gallic acid itself is signaling to the fungus, you don't need to make this. But regardless, we are now armed with a target to help alleviate this massive, massive problem. And so a project was just launched here at HudsonAlpha with sponsors to identify peanuts that have double the amounts of gallic acid in their seeds, to test them to see if they also are immune to aflatoxin, and to use genetics, the power that we have here, to identify that tiny gene that can have this big effect and help us deploy varieties around the world that can mitigate contamination of aflatoxin and reduce this massive, massive problem for those folks.
You get one too, huh?
I got one.
The mission of HudsonAlpha is to use genomics. Oh, they made the circle very wide for the 2 of us.
You got to fit us both in.
The mission of HudsonAlpha is to use genomics to improve life. That is a tall, grand challenge that we are facing. Being a genomicist is quite a challenging job in some ways, in terms of scale. People like Josh and I, we study molecules like DNA that we can't even see with the naked eye. We study changes and processes that might be years or decades down the road before we know if they really worked or not. Being a genomicist at a place like HudsonAlpha requires hope. Hope in the fact that what we are doing matters for the future of our existence as a species, for the next generation. The idea that the genes that we find at a place like this, it's almost insurmountable scale. There are hundreds of thousands of species of plants on our planet alive today. Each of those species has tens of thousands of genes, and each of those species has hundreds of millions sometimes of individuals that live across the planet. We are searching for the smallest of the smallest of the smallest in the biggest haystack you could ever imagine. That is the grand challenge that we have. At a place like HudsonAlpha, I say it all the time, we are a printing press. We are producing the biggest libraries that allow us to search for the smallest word in the greatest library of all time. For us, the power of an institute like this is for people like Josh and I to find those tiny genes that can have big effects.
And so, remember I asked you if you would believe me if I told you that a single gene could help save 1 billion lives. Well, in the 1960s, the world was facing famine. The population had grown to the point where we didn't think we would be able to feed, well, not me, I wasn't around then. Experts didn't think we would be able to feed that growing population. And there was a young PhD plant breeder who was working in Mexico who had an idea. He saw a variety of wheat that was identified in Japan that had one single mutation that caused it to be a dwarf wheat. Now, those of you that know something about wheat know that it used to be quite taller, and it was susceptible to falling over by wind and rain, and it would rot in the field. And it would take so much energy to grow tall that it wouldn't put that much energy into the grain. And so this young researcher thought, what if I take this single gene and I put it in a variety that's resistant, that's already pretty high yielding, and I create a shorter wheat variety that can resist that lodging? Could we feed this growing population? And that's what he did. And those dwarf wheat varieties were shared around the world, and in less than a decade, yields doubled in India, in the Middle East, and in Mexico. And that one man and that one gene, not the one man, a lot of people worked with them, but the one gene helped save 1 billion lives.
Thank you all so much. Josh and I really appreciate all of you coming out today to support the work and the science that we do here at Hudson Alpha. Bob and Sandy Hovde, we're especially grateful for the two of you for sponsoring this event tonight. And Elizabeth Herron, I'd like to pass it off to you.
Thank you, Josh. Thank you, Alex. Wow. That was amazing, right? Wow.
You said you were going to do it.
And following the presentation, there'll be a concert? No? Okay. All right. Hi, y'all. I'm Elizabeth Herron, and I have the pleasure of leading the advancement team at Hudson Alpha. And we're just so glad to see so many of y'all here tonight. I echo the thanks to Bob and to Sandy for making this possible. Thank you. And I also want to thank Mark and Evolution. Mark's the presenting sponsor for our anniversary series. This is one of the talks in the anniversary series, so we appreciate all of you. Absolutely. I hope you all enjoyed, I think you did by the response, what you heard tonight, and learned some new information about the plant science program at Hudson Alpha. You know, when Jim and Lonnie started Hudson Alpha, they created a mission to use the power of genomics to apply that to improve health for people around the world. And I've had people who've said, well, we know you do research in human disease, but we didn't know about why you're doing plant research. And I just think that Josh and Alex gave a great explanation as to why our research in plant science is so beautifully tied to our mission to improve health, to use genomics, tiny genes, big effects, to improve health for people all around the planet. You all may know, oh, actually I want to say this too. Alex has said this to me and I've repeated it several times, that if you can change a plant, you can literally change the world. You can impact billions of lives, because the food that we eat, the water we drink, the air we breathe, what more could improve or affect health than those things? And that's exactly what these two gentlemen and others in our plant science team are working on. So I'm sure many of you know that Hudson Alpha is a nonprofit. We're a 501c3 organization, and we depend on donations from individuals, corporations, foundations, and also from grants to keep us going, to help fund the important work that these gentlemen are doing. So I ask you to please consider, if not tonight, then maybe sometime in the future, and I think there's going to be a slide. There? Okay, it's good. So there's a QR code behind me as a way to go on our website where you could make a donation to Hudson Alpha. And do that in support of the work that these two gentlemen are doing for our plant science team. I also want to point out Alyssa Makowitz and Jeff Moore, who are also on the advancement team, who are standing back over here. They'd be happy to talk to you. You might think about making a donation and name a tree on our campus. You can do that, and we have lots of trees. Or perhaps a planter in our new, where are they, Herb and Terry Lewis demonstration garden that we're going to have at the greenhouse. Or maybe something more like supporting one of these faculty investigators and their teams. There are many options to get involved, and we'd be very happy to talk with you about that. Either Josh, Alex, Jeff, Alyssa, me. Rick is here, Neil is here, Danny's back there. There are lots of us here that would be very happy to talk with you. We'd also like you to know that while we're delighted you're here tonight, if you haven't had a tour of Hudson Alpha, if you haven't seen what we do here, we'd be glad to help set up a tour for you. We would love to have you get our Engage newsletter and stay connected with us so that we can invite you to future events. And I think Alyssa has some sign-up sheets there before you leave at the security desk, where if you just give us your name and best way to get in touch with you, then we can make sure you're added to our list. So with that, I will say a very big thank you. Thank you to you guys. Thank you to all of you who are here. Thank you for the many ways that you engage with Hudson Alpha and the ways you support us. We wish you a very pleasant evening and hope to see you again on our campus soon. Good night.